Dear friends, welcome to Biology Made Easy. Today we are going to discuss about RNA. You know that both DNA and RNA are nucleic acids. They are long chains of polynucleotides. But the difference between DNA and RNA is DNA is double stranded while RNA is single stranded. Now during the course of evolution, it is thought that perhaps RNA evolved first, then DNA evolved after certain modification. Now, just like DNA, the RNA also consists of nitrogen base, sugar and phosphoric acid. But here the nitrogen bases are adenine, guanine, cytosine and uracil. But in case of DNA, it was thymine. And the sugar is ribose sugar. And the phosphoric acid is there. So, just like DNA, the ribose sugar and the phosphoric acid, they form the backbone of RNA, while the nitrogen bases, they project inside. And the number of nucleotides in one RNA usually ranges from 80 to 12,000. And now this RNA can function in different ways. It may act as a genetic material whenever there is no DNA, but its main function is non-genetic. If we will discuss how they are classified, then you will see that it can be classified as genetic material or non-genetic material. Now the genetic material, again it may be single-stranded RNA. RNA is usually single-stranded, but in some viruses you can get double-stranded RNA. But the non-genetic RNA, it is again of different types. One is rRNA, second one is mRNA, third one is tRNA, and fourth one we can write others. In others again we can get different types. It may be snRNA, small nuclear RNA, scRNA, small cytoplasmic RNA, hnRNA, heterogeneous nuclear RNA. We can get antisense RNA. We will come back with all these types of RNA during the course of our discussion. But the main point of discussion here is these three types of RNA, rRNA, mRNA, tRNA. Because these are the three types of RNA which have main function. So now let us discuss one by one. Now first we'll discuss the rRNA. rRNA means ribosomal RNA. rRNA. So it means ribosomal RNA. It constitutes about 80% of the total cellular RNA. That is why it is most abundant. It is also very stable. That means originally it cannot break. It is stable. Then it is synthesized inside the nucleolus. And inside the nucleus, how it is synthesized? Because the nucleus is attached to SAT chromosome. And the SAT chromosome, there is one ANOR part, or secondary constriction, where there is rDNA, and that rDNA synthesizes rRNA. So this is that rRNA. Now this rRNA is again of different types in case of eukaryotes and prokaryotes. In case of eukaryotes, we know that the ribosome is 80S. The 80S again of two subunits. One is 60S, the other is 40S. And in 60S, the RNA are 28S, and in the smaller unit, it will be 18S, 5.8S, and 5S. That means there are four types, and all these are rRNAs, four types. Similarly, if you come to prokaryote 70S, the two subunits are 50S and 30S. And in 50S it is 23S rRNA, here it is 16S and 5S. Now the 28S which is present in the bigger unit of eukaryotes and the 23S which is present in the bigger unit of the prokaryotes, they have enzymatic activity which is very important for examination point of view. Because when protein synthesis takes place, that protein synthesis takes place in the bigger unit. All the amino acids are joined together by peptide bonds in the bigger unit. And in that bigger unit, the peptide transfer enzyme is nothing. They are these ribozymes. So from that point of view, it is very important. Now, this rRNA, it also plays very important role during the 6 kingdom system of a classification. And in RNA, we can get more content of GC than that of AT. And it is very long, and it's so long, sometimes it may be coiled. But even if it is coiled, the stability is not destroyed. Another very, very important thing. During protein synthesis, what happens? Suppose this is the mRNA, this is the 5' and this is the 3'. Now, the smaller unit of the ribosome will be attached here. So, in the smaller unit, what is there? In case of eukaryotes, 18S is there, and in case of prokaryotes, 16S is there. That means in eukaryotes, 18S rRNA, that has complementary sequences to the initial sequences of mRNA so that it can bind easily. That is very important. Now, let us come to the second type of RNA, that is mRNA. This rRNA, it is also transcribed from the DNA by means of RNA polymerase 1. Now, mRNA constitutes only about 3 to 5% of the total cellular RNA. It is quite unstable and it is transcribed from the DNA by RNA polymerase 2. Very important from examination point of view. Now, the mRNA lifespan is very short. In case of prokaryotes, it is only 2 minutes. And eukaryotes, it is only 4 hours. That is why it has to complete its work within time. So, when this mRNA comes from the DNA after transcription, this is the 5' side, this is the 3' side. In case of prokaryotes, at the 5' side, we'll get a Dalgarno sequence. Dalgarno sequence after the name of the scientist, where you will get A, G, G, A, G, G, U. And on the other side, we'll get polyadenylation. And this mRNA in case of prokaryotes is usually polycistronic. Polycistronic means one single mRNA can synthesize so many types of proteins and enzymes. And the glaring example is lactoperon. In lactoperon there are 3 genes. In histidine synthesis, there are 10 genes. That means it is polycistronic. But the eukaryote mRNA is always monocistronic. In case of eukaryotes, in the 5' side, there is a cap, that is 7-methylated guanosine. And on the 3' side, there is polyadenylation. Again, inner to this 5', there is one region which is known as UTR. And similarly, inner to this 3', there is one region known as UTR, untranslated region. That means the nucleotides which are present in those regions, they will not be translated into proteins. Why they are present? For efficiency of protein synthesis. Then in between these two, there are several nitrogen bases, and the conjugative three, they are known as codons. And according to genetic code, each codon specifies one amino acid. So this is the structure of the mRNA. Now let us come to the third one, that is transfer RNA. Transfer RNA is also known as adapter RNA or soluble RNA, and it constitutes about 15% of the total cellular RNA. It is the smallest RNA, having 80 nucleotides, and size is only 4s, so small in size. Now, the tRNA is transcribed from DNA by RNA polymerase 3. That means you can remember RNA polymerase 1, 2, 3 RMT. 1 is for rRNA, RNA 2 is for mRNA, and 3 is for tRNA. 1, 2, 3 RMT. Now, the structure of the tRNA is very interesting. The Robert Hulley gave one two-dimensional secondary structure which is known as cloverleaf model. It is a very famous model. But in three-dimensional structure and tertiary structure given by Kim, it looks like inverted L shaped. Inverted L shaped like this. Three-dimensional, and in two-dimensional it gives a cloverleaf structure. So how this cloverleaf structure looks like, let us see. That is very important from examination point of view. This is the famous cloverleaf model given by Robert Hulley. You see, a single strand of RNA is coiled like this. This is the 5' and this is 3', and all these nucleotides can be paired, but still the stability is not destroyed. Now, in this structure, there are 4 different loops. This loop, where the nucleotides are unpaired, this is known as a T size C. This is known as variable loop. This is known as anticodon loop, and this is known as a DHU loop. They have different functions. In the 3' end, CCA are added, to which the amino acid is attached. Because the main function of tRNA is to carry amino acids from cytoplasm to the ribosome at the time of protein synthesis. So this amino acid is attached in this 3' site. Now in this T site, this is ribosome recognition site. To which ribosome that amino acid will go? That will be recognized there. This anticodon site, this will recognize the codon, because the codon of mRNA and the anticodon of tRNA, they will pair with each other. And this DHU loop, this is the amino acid recognition site. It will recognize which amino acid will be attached. That means there are 4 loops and 4 reaction center. And this variable loop, what its function is not known. One student asked me, that in my book, this T-C loop is given in the left side and DHU is given in the right side. But you are telling that this is in the right side, this is left side, which is correct? I told you are correct and I am also correct. Then he asked how? I told him, you see your book. If this side T-C loop is given, then this must be 3 prime, that must be 5 prime, and the variable loop must be in this side. Then next day, he came with a book and showed that, sir, what you have told is actually correct. That means my aim of telling this is the T-C loop always lies in the 3 prime side and the DH loop always lies in the 3 prime side. That is important. Now, if you mark another very important thing, here T-C, T means thymidine, but thymidine is not present in case of RNA. So this thymidine is ribothiamidine. Ribothiamidine. This cytosine is pseudocytosine. This DHU is dihydroxyuridine. And here in the anticodon, sometimes we can get inosine. Now these bases are unusual bases present in tRNA. This is very, very important from examination point of view. Unusual bases. Now why these unusual bases are present in tRNA? These presence of these unusual bases make the tRNA coiled structure and a stable structure like this. So it helps in its function. So these are the important functions of rRNA, mRNA and tRNA. We will come to the other types of RNA which we have discussed in the beginning during classification. The snRNA is known as small nuclear RNA that is present in nucleus. The scRNA is a cytoplasmic RNA present in cytoplasm. And the hnRNA, during transcription of mRNA, that first the hnRNA is synthesized, then it is converted up due to splicing to mRNA. And antisense RNA, that is a complementary sequence to the RNA, and the details of all those things we will discuss during our regular classes. So this is all about RNA. Now I will give you few questions. Now number one question, how many types of rRNA are present in eukaryotes? A choice 1, B choice 2, C choice 3, D choice 4. Then the second question, which RNA is abundant in cell? A choice rRNA, B choice mRNA, C choice tRNA, D choice snRNA. Now the third question. In the third question, you can tell which RNA has enzymatic activity, and this same four choices, rRNA, B choice mRNA, C choice tRNA, and D choice snRNA. So after going through the video, you will be able to answer all these questions. So thank you very much for watching it and have a good day.